Hello, I'm Flagg Flanagan, Chairman and CEO here at DiscGenics. This is a very exciting time in our business. We are currently conducting regulator-allowed human clinical trials in the United States and Japan for our first product candidate, IDCT, which is a homologous and allogeneic injectable disc cell therapy for degenerative disc disease. At the same time, we developed phase 1 manufacturing processes to support our current clinical efforts. Now, in preparation for an eventual commercial program, we are in the process of setting up our own large-scale manufacturing facility here in Salt Lake City. When this scale-up is complete, we will be able to generate thousands of doses of IDCT per lot to the highest global cell manufacturing standards.
My name is Lara Silverman. I'm the Senior Director of R&D here at DiscGenics. Today, we want to give you a glimpse into our research labs, where our team is working hard to improve and scale up the manufacturing process to generate IDCT in a GMP-compliant manner. We begin by procuring adult intervertebral disc tissue. The tissue is obtained from carefully selected donors and processed by our highly trained scientists. Cells derived from the disc tissue are grown in specialized conditions to generate the unique cell phenotype needed for IDCT to be active. The cells are being converted into the therapeutic progenitor cells that we noted were effective in animal models of disc degeneration. Here you can see individual cells growing and dividing into more cells over the course of 5 days. We're able to take measurements and counts of the cells to monitor growth and phenotype. Halfway through the process, we freeze the cells in the vapor phase of liquid nitrogen, which is negative 196 degrees centigrade. Freezing the cells gives us enough time to perform rigorous testing to ensure the safety and identity of the cells. After the cells pass the required testing, they are thawed and further expanded, this time using larger systems that allow for the generation of significantly more cells. During this time, we do in-process analytical testing to ensure the cells are growing properly. We also grow the cells in a bioreactor, currently in a small volume, but eventually in very large volumes in order to generate the many doses we anticipate we'll need to meet market demand. In this bioreactor, we are able to control variables such as temperature, pH, and oxygen in order to generate the optimal conditions for cell health, growth, and behavior. When the process is complete, we freeze the cells in their final form and perform release tests to evaluate the purity, potency, identity, and safety of our product. Using state-of-the-art equipment, we can evaluate these properties and ensure they meet preset criteria that we know is associated with clinical safety and efficacy for this homologous and allogeneic cell therapy. Each of these steps is critical to ensuring we are upholding the rigorous clinical safety and manufacturing consistency standards placed on us as developers of an IND-approved BLA pathway product. It is also what sets IDCT apart from minimally manipulated products that are available in the marketplace, yet have limited process controls and fewer analytical and clinical testing protocols. The final product is a disc cell therapy for degenerative disc disease, which is off the shelf. It's delivered to the patient via injection in a procedure room. As Flagg mentioned, we're currently executing regulator-allowed, double-blind, vehicle and placebo-controlled clinical studies in both the US and Japan. Once we demonstrate safety and efficacy of IDCT, we intend to approach the regulators with a biologic license application, which is a BLA in the US, and a conditional marketing authorization, which is a CMA in Japan. The steps we've taken to adhere to the many regulations of a manufactured cell therapy are plentiful, but we believe this is the best and only way to bring a product to the market that promotes patient safety and quality healthcare.
I hope you've enjoyed this look into our facility here at DiscGenics. We are very proud of the amazing, hard-working team here in Utah, collaborating and innovating daily to bring this cell therapy to patients safely and effectively. IDCT has the potential to help millions of patients worldwide, and we are honored to be stewards of this game-changing medical innovation. As is our vision here at DiscGenics, imagine all the people we can help.